All right, welcome back. So this week, as you can see, we're moving on to the urinary system. Now, in saying that we're talking about the urinary system, primarily we're going to be talking about the kidneys because I want you to understand the important filtration function that the kidneys serve. We're not really going to get into the ureters or the bladder or the genitalia or any of that kind of stuff. Basically, this is, even though it says the urinary system, we're primarily talking about the kidneys. And the kidneys do generate urine. But again, my primary interest is that you're able to see the really important role that the kidneys play in performing a variety of functions, whether that is helping us to maintain blood volume and thereby blood pressure, or helping us get rid of waste products, things like urea. So they play, like I said, a really important role in filtration and maintenance of blood pressure. So I figured it was worth talking about in this course. So on a related note, in terms of what the kidneys do, so they serve a vital role in first maintaining water balance. And so by determining how much water we either hang on to or excrete, they affect plasma volume. And remember that plasma is the fluid component of the blood. And so if we get rid of a bunch of plasma, well, then what we're going to do is decrease blood volume because you're losing the fluid component of the blood. And so the blood effectively then gets thicker. Or if we hang on to too much plasma, then the blood gets thinner, right? So you have a greater volume of fluid that the red blood cells are floating around in. But what that does then is it creates a higher overall blood pressure by having a greater blood volume in the system. So they help us to maintain water balance, but by doing that, like I said, they help us to maintain blood pressure within narrow limits, relatively narrow limits at rest. And then, so other things that the kidneys do, so they help us maintain concentrations of ions. So remember that ions are electrically charged particles, so things like sodium, potassium, calcium, chloride, they help us to maintain those concentrations in extracellular fluid, so fluid outside of the cells. So the reason that that's important, one of the reasons that that's important is fluctuations in those can be fatal. So we talked previously about hyponatremia, so the condition where basically by excessive water consumption you can flush out too much sodium from the extracellular fluid, and by doing that you can cause swelling inside the central nervous system. So that's a throwing off of the concentration of ions. Similarly, if you accumulate excess potassium in the extracellular fluid, that's going to throw off the function of the nervous system, and that could also be fatal. So we have to maintain those extracellular ions in a relatively fine limit there, and so the kidneys play a really important role in making sure we don't normally get rid of too much sodium, or we don't hang on to too much sodium, we don't get rid of too much potassium or hang on to too much, etc., right? So they help us to maintain that balance. And then they also help us to maintain our acid-base balance. So hopefully you'll remember that the blood is slightly basic. So we don't want the blood to get too acidic. And so one of the things that we can do is to secrete, in this case, excrete, but also secrete is how the kidneys actually do it, get rid of extra hydrogen ions. And so by getting rid of those hydrogen ions, we can maintain that property of the blood that it is slightly basic. The other thing that the kidneys do is that they hang on to, you'll see HCO3 minus in the text and in some of the graphics. That's bicarbonate. So they help us to hang on to bicarbonate, which serves a really important buffering role in the blood, and then also plays a role in the transport of carbon dioxide in the blood as well. Obviously, one of the really important functions of the kidneys is to get rid of metabolic waste. So the primary waste that they get rid of is something called urea. Well, they get rid of ammonia and urea. And so those are byproducts of breaking down proteins. So whenever we break down proteins, we have to get rid of nitrogen. And so the kidneys help us to do that. So urea is a byproduct of that protein breakdown. So the kidneys help excrete that. But in addition to helping to excrete protein, the kidneys also help us get rid of creatinine, which is a breakdown or formed from the breakdown of creatine phosphate in the muscles. They help us to get rid of uric acid, which is formed whenever we break down nucleic acids. So in saying nucleic acids, things like DNA and RNA. So there's some turnover in those nucleic acids. Obviously, we're making lots of copies of DNA when we're making red blood cells and other cells that turn over really quickly. So there are some waste products that are formed, and so uric acid being the waste product in that case. And so then we have to get rid of those. And so the kidneys play a role in excretion of that. They also produce EPO. So EPO stands for erythropoietin. We talked about that in the blood chapter. So erythropoietin is an important stimulator of red blood cell production. So remember that we talked about it in the context of blood doping, of basically cheating in sports. And so if you take EPO, like exogenously, if you actually just take the hormone, that is going to cause you to make additional red blood cells. If you make additional red blood cells, then you can transport more oxygen. So again, viewing this from a sports standpoint, you can transport more oxygen. Because you can transport more oxygen, you can work at a higher intensity for longer. And so the kidneys produce EPO. Obviously, they don't do it for sport performance effect. What they do is they monitor saturation of the blood or they monitor blood oxygen saturation. Let's go with that. And so they monitor that. And so if blood oxygen saturation gets too low, then that'll stimulate the production of EPO to make additional red blood cells. So that saturation may be the result of things like blood losses, or it could be the result of doing something like going to altitude. And so whenever you do that, one of the adaptations to being in altitude long term, so over the course of several months, is that you're going to make additional red blood cells. And so it's the kidneys that are responsible for detecting that reduced oxygen saturation in the blood, generating EPO, which in turn then causes us to generate more red blood cells. The kidneys also convert vitamin D to its active form. And the reason that matters is that's going to help us to play a role, or that will play a role in maintaining calcium homeostasis. So remember that you need vitamin D in order to absorb calcium. So that's going to be an important function for them. And then the last thing on there is that they can perform gluconeogenesis. What is that? That's making glucose from proteins. So gluconeogenesis is something that we do during fasting. So we do it actually primarily in the liver. So for example, if you've heard that you should not work out fasted, because sometimes people talk about making sure you eat before you exercise, or you want to minimize how long you go between meals. The idea there is that if we fast for long enough, that we deplete our liver glycogen stores, so our liver sugar storage. Yeah, our liver sugar stores. Let's go with that. If we fast for long enough, then we deplete those stores. And so what we have to do is to make additional sugar, which we can do from amino acids, which are the building blocks of proteins. And so the primary amino acid that we do that from, or the amino acid that we do that from, is glutamate. And so again, primarily we do that in the liver, but the kidneys can also do that under conditions of really prolonged fasting. So that's an important function that they serve as well. So basically to maintain blood glucose, if we haven't eaten in a really long time, they can assist the liver in maintaining additional glucose. So in terms of where they are, so you can see the kidneys there, and you probably noticed that the right one sits lower than the left. So in general, they sit somewhere between T12 and L3. So T12 is your lowest thoracic vertebrae. Remember, your vertebrae are spine bones. L3, so you have 5 lumbar vertebrae. L1, 2, and 3 are the top 3. So they basically sit at the very bottom section of your rib cage and the very top section of your low back. As you can see, the right one sits a little bit lower than the left. And the reason for that is because, and it's not pictured there, but your liver would be right here. And so the liver is kind of triangle shaped. And so because it's larger over here on the right side, that pushes the right kidney down just a little bit. So the right one sits a little bit lower than the left because it's crowded by the liver. And so the top portions of them are protected by the ribs, or the superior portion, to use the more anatomical term. So if you're going to get a laceration or an injury to them, it's typically the lower portions of them. They're fairly small, so each kidney is only about the size of a bar of soap. So I don't know, I pictured them being bigger, but they are fairly small. And they actually do shrink with age. So they end up between the ages of 20 and 80. So I think there's something like 300 grams. I didn't do the conversion on that. But somewhere around 300 grams whenever you're age 20. By the time you're age 80, they drop to around 200 grams. So their size decreases by about a third across the course of the lifespan. So, and as you can imagine, as they decrease in size, their function drops some. And in fact, some you lose, I think it's somewhere in the neighborhood of 40% of the nephrons, which are the functional unit of the kidneys. So they lose somewhere in the neighborhood of half their functional capacity by the time if you're lucky enough to reach age 80. So their function decreases across the lifespan as well. In terms of other anatomical stuff, so there's 3 layers of fascia in and around the kidneys. And so the way they're laid out here is from superficial to deep, so closest to the skin surface to farther from the skin surface. And so the first layer there, the renal fascia. So the job of the renal fascia is to anchor the kidney to the surrounding structures because it's the most superficial of the layers of fascia. So it is dense, irregular connective tissue. And so remember that dense refers to the density of the collagen fibers. So if it's dense connective tissue, that means there's lots and lots of collagen fibers, not a ton of extracellular space. And then irregular means that the collagen fibers go all kinds of different directions, right? As opposed to regular connective tissue, which is where they all run in parallel to each other. So the renal fascia then, dense, irregular connective tissue that helps anchor the kidney to the surrounding structures. Then deep to that, we have the pararenal fat capsule. And so the pararenal fat capsule is basically a cushion for the kidney. So it's a fat layer of fat that helps absorb shock and protect the kidneys from trauma. And then the innermost or the deepest layer is the fibrous capsule, which is also dense, irregular connective tissue. And just like the pararenal fat capsule serves an important function in being a barrier against trauma, but it also helps to maintain kidney shape. So in discussing the pararenal fat capsule, one of the things that struck me there is I probably should have mentioned that anything renal deals with the kidneys, right? So you can see a lot of renal here. So renal artery is an artery that goes to the kidneys, the primary artery. The renal hilum we'll talk about here in a second, renal vein, etc. So anything renal deals with the kidneys. So if somebody's in renal failure, they're in kidney failure. All right, so in terms of the anatomy, we're going to change pictures here. So this is your right kidney frontal cross section, and so you're looking at it, looking at the inside of the right kidney here. So the 2 largest or 2 most obvious demarcations, you have the renal cortex and the renal medulla. So the cortex is the most superficial layer of the kidney. So just like we talked about with the brain, for sure. So remember we talked about the cerebral cortex. And so the cerebral cortex is the outermost layer of the brain, or at least the outermost layer of the cerebral hemispheres. So the cortex is the most superficial layer. And you're actually going to see the same thing when we get to the endocrine system. We'll talk about the adrenal glands. And so the adrenal glands are also going to have a cortex and a medulla. So the cortex is outside or superficial, the medulla is deep. And so that's the same thing here. And so our renal cortex is out here. And then our renal medulla is in here. And so one of the things that you can see is that we've got these little extensions of the cortex here. And so those little extensions of the cortex, those are your renal columns. So the columns then are extensions of the cortex. In the renal medulla, so these guys here, so those are called pyramids. And so just like some of the other pyramid or pyramidal shaped things that we talk about, so like the patella, for example, there's a base and an apex. And so the base is here, it's the wider part of it. The apex is here. So the apexes are deep, the bases are superficial. And so those are our pyramids. I want to make sure I touch on everything here. So one of the things you'll see in terms of kidney anatomy is the idea of lobes of the kidney. So each kidney has approximately 8 lobes. There's some variability between people, but there's approximately 8 lobes to each kidney. And so a lobe includes one of these pyramids of the medulla and then the adjacent columns here. So it's kind of like half of this column and then all of this cortical tissue as well. So each of these things, each of these pyramids plus the columns, and then the adjacent cortical tissue, all of that's a lobe. So here's one lobe here, second lobe here, third lobe here, etc. On down. Other things, so you can see that the most lateral surface of the kidney is convex, so it curves out, and then on the medial surface it is concave. So this area here, that is concave on that medial surface. And on the medial surface is where we get the entrance of the blood vessels, or the exit of the veins in that case. And then you also have the renal pelvis here. And so the renal pelvis is the primary drainage vessel of urine. So all of these things, you see this term here, calyces. So calyces is plural. Singular is calyx. And so you have 2 to 3 major calyces. So here would be one, here would be a second one, and here would be a third one. So those are your 3 major calyces. And then those split off into minor calyces. And so, whoops, go back. And so those then, basically those are, so we'll talk here in a second about collecting ducts. So we're going to have the formation of urine dumps into the collecting ducts, and it goes from the collecting ducts into the minor calyces, into the major calyces, into the renal pelvis, and then eventually you can see that the renal pelvis empties into the ureter, right? And so then the ureter goes down to the bladder and then out beyond that. All right, make sure I got everything I wanted to there. I think I did. Okay, good. So there's all those. Blood supply. I don't need you to know much about blood supply to the kidneys, except that they get a lot of it. So at rest, the kidneys get about 25% of cardiac output. So they get somewhere in the neighborhood of 1.2 liters per minute of blood flow. That obviously depends upon your stature. So the larger you are and the better shape you're in, you tend to have higher blood volume. But you know, in terms of the absolute volume that they get, it's somewhere in the neighborhood of 1.2 liters per minute. If you're smaller, it's a lower number. If you're bigger and in better shape, it's a higher number. But the important part there is that at rest, the kidneys get about 25% of cardiac output. So they're doing lots and lots of filtering at rest. So in terms of the blood vessels, so one of the things you're going to see, so obviously the reds are the arteries. So we're going to get the aorta is going to have a direct 90-degree branch, or the, so you're going to come down off of the aorta, and without getting into too much detail about the blood vessels, the renal artery is going to split off from the abdominal aorta at a 90-degree angle here. And so then, there we go. Got my little pointer back. All right, so we're going to split at a 90-degree angle from there. Then we're going to split into segmental arteries, interlobar arteries, and then arcuate arteries. And so most of that blood flow goes into the cortex. About 90% of the blood flow actually goes into the cortex of the kidney. And then it'll return basically in the opposite fashion. So veins with similar names. So if we've got the interlobar artery, we have the interlobar vein, renal artery, renal vein, etc. Yeah, I think that's all I wanted you to know about the blood flow. So just know that the kidneys at rest get a lot of it because they're constantly filtering, right? So in terms of constantly filtering, so one of the facts that's in the book, I don't know, like 5 or more times in this chapter, is the fact that the kidneys filter somewhere in the neighborhood of 180 liters of fluid per day, but you only actually excrete about 1 to 2 liters. So by saying excrete, you only actually urinate out about 1 to 2 liters per day. But the kidneys are turning over fluid all the time. So another one of the fun facts in the book is that you turn over your plasma volume, give or take, every half hour. So in order to do that, then obviously there's a pretty significant blood supply to the kidneys. All right, so here's where a lot of the questions will come from. Here's the more important stuff I want you to know is more of that microscopic anatomy of the kidneys, so how they actually function. So you can see there that the nephrons are the functional unit of the kidneys. So what does that mean? So a functional unit, when we say that, it's the smallest physical structure capable of carrying out the function of an organ. And so in the case of nephrons, they carry out that filtration function of the kidneys. So that's the smallest individual unit that can do that. And so we have to have both of the primary parts of the nephron in order to be able to perform those filtration functions. So you can see, so as alluded to there, the nephrons have 2 primary parts. So we have the renal corpuscle and then the renal tubule. So the corpuscle itself is broken down into 2 smaller parts. There's the glomerulus and then the glomerular capsule. So the glomerulus, what it is a little tuft of capillaries. And so all of this little tuft of capillaries here, that's our glomerulus. And I think we have a better picture here. So you can see just the capillaries by themselves, right? So that's the glomerulus, this little, like I said, tuft of capillaries. And then the glomerulus extends into this capsule here. That's the glomerular, or the glomerular capsule. I was going to tell you the alternative term there, which is Bowman's capsule, because you'll see both. But I'll try to use the more anatomical term and not scientist name. So we have the glomerulus, tuft of capillaries, the glomerular capsule. And then we have the renal tubule. The tubule has three basic parts to it. So there is the proximal convoluted tubule, which you'll see abbreviated PCT a lot. So there's the proximal convoluted tubule. And so proximal meaning that it's closest to the capsule. And then we have the nephron loop down here. So there's two parts to the nephron loop. We won't get into the thin and thick segments, but one of the things I do want you to know is that there is a descending segment, so the part that goes down, and then there's the loop, and then there's the ascending segment that comes up, because that's going to be important when we talk about filtration. And then on the way back here, we have the distal convoluted tubule here, and then we're going to dump whatever's left still in that filtrate. That's once we dump it into this thing, which is a collecting duct, then that is going to be our urine. And so then that, as mentioned, dumps into the calyces, minor to major, to renal pelvis, and then eventually into the ureter. All right, so let's see, make sure we talked about everything here. In terms of the collecting duct, there's something else I wanted to tell you about that, but I don't remember what. All right, well, moving on anyway. All right, so let's look at it a little bit closer up. So here's the close-up. So up top here, we'll ignore this picture for now. So up top here, this is our little glomerulus, right? So our little tuft of capillaries. And then here, we have our glomerular capsule. So in terms of the glomerulus, things to know about them. So there is an afferent arteriole, and so the afferent arteriole brings in arterial blood, and then the efferent arteriole takes the blood away after it's gone through the glomerulus. One of the things that's important about those two structures is the fact that both of those are arterioles, and again, why that matters is that because they're arterioles, there is smooth muscle around them, and so that smooth muscle is able to constrict a little bit. And so by being able to constrict, that's going to help maintain pressure inside of the glomerulus, which plays a really important role in ensuring continued filtration. The other thing is that the efferent arteriole, and you can kind of see it in this diagram here, but the efferent arteriole is slightly smaller than the afferent, so the passageway coming in is larger than the passageway going out, and so that is another important structural adaptation that helps us to maintain pressure here in the glomerulus. The higher the pressure is here, the more it's going to push fluid out of this blood, so the more it's going to push plasma out of this blood and into the glomerular capsule. So basically what happens here is that on the afferent side, we're going to then run blood through the glomerulus, so run it through this just torturous labyrinth of capillaries. And so when we do that under pressure, that's going to push the plasma out of the blood. Not all of it, but a lot of it. And so when we push that plasma through through that filtration membrane, the vast majority of what goes out is primarily plasma. So actually, we'll talk about the filtration membrane here in a second, but for the most part, proteins can't escape. So proteins are pretty big, and so proteins get caught up in this filtration membrane, so they end up staying in the plasma. Obviously, your red blood cells are pretty big, so they can't make it through the filtration membrane. So what ends up leaving then, which any of the fluid that leaves the glomerulus is called filtrate. So what ends up leaving the glomerulus and becoming filtrate is primarily plasma, so the fluid component of the blood. But in addition to that, it's going to take with it things like individual amino acids, so not whole proteins, but some of the building blocks of proteins because they're pretty small. It's also going to take glucose with it, and then it's also going to take a lot of your ions, so things like sodium, potassium, calcium, those are going to leave as well. And so then that's the filtrate. And then we have our paratubular capillaries and then the vasa recta. And so the vasa recta is down here, and then here's our little paratubular capillaries here. And so both the paratubular capillaries and the vasa recta are going to supply different parts of the tubule with blood, or not blood, they're going to supply them with oxygen, right? So they're going to supply the nutrients that the cells, because a lot of these are cuboidal cells, there's some squamous cells, depending on which part you're talking about, but those cells still need nutrients, and so the peritubular capillaries are going to supply a lot of this. So you can see this is the proximal convoluted tubule here. So the peritubular capillaries are going to supply those cells with the nutrients that they need, and then for the most part, the vasa recta does these long descending loops here, and so it's going to supply those cells with the nutrients that they need. But both of those structures also perform a really important reabsorption role. So again, having said, the fun fact the book uses that we produce, or the kidneys filter 180 liters of fluid per day, but we only get rid of, or we only excrete about 1 to 2 liters as urine, that means then that we have to reabsorb almost everything. And so that reabsorption occurs in the peritubular capillaries, but also in the vasa recta. And so they serve a really important reabsorption function. So the juxtaglomerular complex, there we go. So in terms of breaking down the word, so if anything is juxta, it's next to, right? And so like if you juxtapose something, you put two ideas next to each other. And so the juxtaglomerular complex then is next to the glomerulus. And so again, the glomerulus, tuft of capillaries, and so the juxtaglomerular complex is here. And so what it is part of the ascending loop of the tubule. So the tubule goes down, goes down, comes back up, and so here is our ascending loop that is actually going to adjoin to both the efferent arteriole and the afferent arteriole. And so there's 3 types of cells that form the juxtaglomerular complex. So the first one here are these macula densa cells. And so the macula densa cells are actually part of that ascending tubule. And so what they do, I was going to tell you the cell type, but it doesn't matter. What they do is they have sodium monitors in them. So they're able to monitor the sodium concentration in this filtrate. And so whenever, depending upon the sodium concentration, if it gets too high, then they can affect blood flow through the afferent arteriole. Or if it gets too low, they can affect blood flow through the afferent arteriole. And we'll talk more about that here in a second. But the thing to know for now about the macula densa cells is that they have sodium concentration receptors in them, so they can alter blood flow into the glomerulus. Then the next one we have are the granular cells, and so those are, you can see, trying to get my mouse back, those are in the little arterial wall. So here's some granular cells here, here's some granular cells here. And so they do a couple different things. One of the things that they do is that they have renin in them. And so renin is also going to have an effect on blood flow through the glomerulus. And in addition to that, they also act as mechanoreceptors. So if there's too much stretch here, if the pressure is too high, then they can oppose that pressure. Or if the pressure is too low, they can go the other way. And like I said, we'll talk more about that in a second. So those are the granular cells, and then there are the mesanglial cells, and so they actually sit between the two. And so the mesanglial cells kind of sit between the granular cells and the macula densa cells, and so they play an important role in helping those two communicate. So ultimately, what the juxtaglomerular complex does is to regulate the rate of blood flow through the glomerulus. If blood flows too fast through the glomerulus, or not too fast necessarily, but if the pressure is too high, then that's going to push too much fluid into the capsule, and so we're going to get rid of too much fluid. So that's why, bringing it back a little bit farther, that's why that matters. So if the pressure is too high, we're going to get rid of too much fluid, and so we'll dehydrate. If the pressure is too low, we're not going to filter enough, and then we're not going to get rid of enough of the byproducts that we need to, and so that's problematic as well. So as with anything else, there's this nice happy medium homeostatic spot in terms of pressure inside of the glomerulus that we need in order to properly filter. And so the juxtaglomerular complex then plays a really key role in making sure that we maintain that pressure, not too high, not too low, so we can maintain an appropriate filtration rate. Speaking of filtration, so in terms of urine production then, there's basically 3 parts to it. So the first is glomerular filtration. So that is when fluid leaves the glomerulus. And then, so we got lots and lots of fluid that's going to leave, but of course we have to reabsorb almost everything. So we reabsorb about 99% of what is filtered out. So we reabsorb the vast majority of the water, sodium, potassium, etc. But there are some things that we don't reabsorb. So obviously we don't reabsorb all of the water or all of the sodium. And then secretion is actually moving things from the blood into that collecting duct. So that we can get rid of specific things. So things that we actually secrete include hydrogen ions, so to help us maintain blood pH. We also secrete, I just had a list of them. Sorry. So I don't know, we'll get to it on a later slide. I have a bunch of examples. But for now, let's go. Oh yeah, so you secrete different drugs and toxins as well. That's one of the ways that we get rid of those is actually through secretion. So we intentionally move them into that filtrate, which then becomes the urine. All right, so I mentioned the filtration membrane a minute ago. So obviously there's your glomerulus, there's your glomerular capsule. And so the membrane itself has 3 layers to it. So there's the fenestrated membrane of the endothelium. So what is that? So remember that the endothelium is the inner lining of a blood vessel. And we say that the endothelium is fenestrated. What that means is it kind of looks like Swiss cheese. There's a bunch of little holes in it, and I thought I had a picture. There's definitely a diagram in the book that shows what that looks like. But basically the inner membrane of all of these little capillaries kind of looks like Swiss cheese. There's a bunch of holes in those cells. As opposed to, if we think about the capillaries in muscle cells, remember we talked about those being continuous capillaries. There's no holes in those cells, and those cells are joined together by tight junctions. In the case of these little glomeruli, though, we want a lot of fluid to be able to move across those cells, and so they actually just have holes in them. So that's what this means. So there's a bunch of holes in the lining of the glomerulus, or yeah, let's go with that. And so because of that, that's going to allow mostly fluid, but also, like I said, some things like glucose and your ions to pass from the bloodstream into the capsule. But superficial to that, there's a basement membrane. And so the basement membrane is going to essentially catch some of the larger proteins. So anything that has managed to squeeze its way through these fenestrations, but is still a fairly large molecule, like some of the proteins, can get snagged by the basement membrane. So the basement membrane sort of plays a backstop to those holes and helps catch some of those proteins. And then anything that wasn't caught by the basement membrane, in terms of large molecules like proteins, gets caught by the foot processes of these podocytes. So one of the things that the book talks about in terms of laying out the structure of the glomerulus versus the capsule. So it actually talks about it being like a 2-layered membrane, kind of like a serous membrane. So the serous membrane we talked about near the beginning of the course. The serous membrane, remember that you have inside of your rib cage, you have one layer of serous cells that lines the inside of your rib cage, and then another layer of serous cells that lines the outside of your lung. And so both of those layers secrete, in that case, a lubricating fluid that helps the lungs slide inside of the rib cage. And so in terms of the serous membrane, there's the parietal and visceral layers. So the parietal layer lines the inside of the rib cage. The visceral layer lines the outside of the lungs. So anything visceral, remember, deals with an organ. And so in terms of parietal versus visceral here, so the visceral layer of the glomerulus is the layer that actually attaches to the blood vessels, and then the parietal layer is out here. And so you can see, oh, I didn't even realize. So it says parietal layer here. There you go. So the parietal layer is these simple squamous cells that help form the glomerular capsule. So that's how they describe it as a 2-layer membrane. So there's a layer of the membrane that actually attaches to the blood vessels, and then there's another layer of the membrane that actually forms the capsule, and then there's a fluid space in between. So in this case, that fluid is called filtrate, as opposed to the serous fluid that I just mentioned with the lungs. So why any of that matters? So the visceral layer is formed by these cells called podocytes. And so the podocytes, you can see they have your little cell body here with its nucleus, but they have these extensions that are called feet. And so you can see the different podocytes interact with each other, where their feet don't quite overlap, but they kind of interlock like puzzle pieces. And so there are gaps between them, but then there's a membrane between those gaps. So those are called the filtration slits, the gaps between, and then there's a membrane that sits on top of that. And so what happens there is any of those even smaller proteins that have managed to make it through the basement membrane, those get caught up in the foot processes of the podocytes, and then they get kicked back into the bloodstream. Oh, here's our fenestrations. I should look at my illustrations better before I start. So here's my little Swiss cheese holes in the endothelium, and then the basement membrane looks like it's not pictured, but that's okay. So why all that matters? That those 3 layers of the filtration membrane allow things, again, primarily fluid out, but also ions and glucose, but they keep proteins, so not amino acids, not the building blocks of proteins, but they keep fully formed proteins from moving out of the blood vessels, so they keep those in there. So one of the things that the book refers to repeatedly is that the filtrate is almost protein-free because of these 3 layers of the filtration membrane. All right, so in terms of the rate of filtration, so there's basically one big outward pressure forcing fluid out of the glomerulus, and then 2 inward pressures resisting that outward push. So the outward push, trying to kick fluid out of the glomerulus and into the glomerular capsule, is hydrostatic pressure. And basically what this is blood pressure inside of the glomerulus, or inside of those capillaries. So you could think of it then as glomerular blood pressure. So that's fairly large. So that is 55 millimeters of mercury. So if you think about your normal systolic blood pressure at rest, that's 120 millimeters of mercury. So as you move from larger blood vessels and move more distally, that pressure obviously drops. In most of your capillaries, that pressure is about 26 millimeters of mercury. And so you can see that it's quite a bit higher in the capillaries of the kidneys. And so why is that? Well, the primary reason is what I mentioned earlier, that this efferent arteriole is much smaller than the afferent arteriole. And so because of that, you got high pressure coming in and then a small blood vessel leaving. And so basically that high pressure kind of sticks around here, can't really exit. And so then that helps force fluid out of the glomerulus. It helps in the creation of filtrate. So that's the outward pressure. And then there's 2 inward pressures. One of them is really similar. So you got the hydrostatic pressure in the capsule. And so what that means is as this fluid accumulates here in the capsule, it's going to push back in its own right. So it has its own pressure pushing back against that hydrostatic pressure coming from the glomerulus. So the outward pressure then is 55 millimeters of mercury. The inward pressure from the fluid in the capsule is fairly small. It's only 15 millimeters of mercury. And then the other inward pressure is this colloid osmotic pressure. So for this, you got to think all the way back to chapter 3 when we talked about osmosis. So remember that, and so osmosis is passive movement of water. So remember that water wants to move through a membrane to balance out the concentration of fluids. And so what we end up with in the glomerulus is, remember, if we're hanging on to all these proteins, if they can't make it through that filtration membrane, then you're going to have a lot of fairly large molecules that end up, especially kind of up in here, on their way out into the efferent arteriole. You're going to end up with lots of large molecules in this remaining blood, and so because of that, water actually wants to move back into the glomerulus to try to balance out the concentration of solutions. It doesn't want to move to where there aren't big molecules. It actually wants to move to where there are. And so that's going to be a force resisting flow as well. And so that accounts for about 30 millimeters of mercury. So if we have our 55 millimeters of mercury pushing out and then 30 pushing back in due to that osmotic pressure, that basically sucking pressure back into the glomerulus created by the concentration of proteins in there, plus that hydrostatic pressure pushing back from the fluid accumulation here in the glomerulus, that accounts for about 15 millimeters of mercury. What we're left then is a net filtration pressure, so out plus in, or out minus in, out minus in, with about 10 millimeters of mercury. So the net filtration pressure is accounting for outward forces and inward forces. Which way is the fluid going to flow? And so the pressure pushing plasma out of the glomerulus and into the capsule is about 10 millimeters of mercury. That's the pressure differential that allows for that fluid to move one way. And so why that matters is you don't really have much of a margin for error. So if that net filtration pressure drops to 0, well, then we stop filtering blood, right? And so that's problematic. So what we have to do then is to maintain this pressure, the outward pressure, fairly normally, because these two don't change much. So we have to be able to maintain the outward pressure fairly consistently. So we have to maintain pretty high pressure here in the afferent arteriole and then into the glomerulus. And so the next few slides are descriptions of how we do that. So the glomerular filtration rate, as you can see, is the volume of filtrate formed each minute by all of the glomeruli of the kidneys together. And so it is proportional to that net filtration pressure that we talked about. So obviously if that net filtration pressure goes down, so if it drops to 5 or 0, then glomerular filtration rate also drops to 0. It's also proportional to the surface area available for filtration. So I mentioned earlier that that kidney function declines across the lifespan, so the kidneys overall shrink, but the number of nephrons that you have decreases as well. And so as that happens, then you have less total surface area available for filtration. And so if we have less surface area available, then our glomerular filtration rate goes down. So as we age, we get less, you get a decreased glomerular filtration rate in the kidneys. You can't filter as much blood, basically, as you age, in part because you've lost you've also lost glomeruli, but you've lost nephrons. And then the membrane permeability, that doesn't really change either acutely or across the lifespan. The filtration membrane its permeability stays pretty constant. So one of the things that we have to do then is to maintain our glomerular filtration rate, and we do that primarily by maintaining that net filtration. So there's a couple different ways to make that happen, to maintain GFR. And so the first one is through intrinsic control, so intrinsic meaning inside of the kidneys. And so the first is a myogenic mechanism. So myo is muscle, genic is originating with. So the myogenic mechanism, what ends up happening there is that this smooth muscle around the arterioles, if it ends up being stretched, then it's actually going to cause a reflexive constriction of those blood vessels. So effectively what's going to happen here is, let's say during exercise, for example. So as you start to exercise, and I'm thinking here of cardiovascular exercise, what you're going to get is an increase in blood pressure. And so then as blood pressure goes up, that is then going to stretch this afferent arteriole, which then stimulates the smooth muscle around it to constrict. Because again, if we increase pressure too much, that's actually going to speed up filtration and probably speed up filtration too much. And why that matters is if we start pushing fluid through here too fast, then it ends up basically, we don't have enough time to reabsorb the fluid that we need to, but we also don't have enough time to reabsorb things like sodium and potassium if we need to. So if the fluid flows too fast, we can't reabsorb everything that we need to, and that's a problem. And so what we want to do is keep the pressure from getting too high, because that interferes with reabsorption. And so the myogenic mechanism then is, as blood pressure increases, we're going to get a stretching of the smooth muscle around the afferent arteriole, which then reflexively constricts to, I'm trying to bring my little arrow back, to close the diameter of this blood vessel, so it slows blood flow into the glomerulus, which then slows down the filtration rate. So the myogenic mechanism then keeps blood flow or pressure from becoming too high in the glomerulus so that we don't increase GFR too much. And then the tubuloglomerular feedback mechanism. So remember, this is going to involve the juxtaglomerular complex. So blood flow in, blood flow out, and then here's our little ascending part of the tubule. So key here are going to be these macula densa cells. So I mentioned earlier that those have receptors in them that detect the concentration of sodium. And so if sodium concentration gets too high in that ascending part of the loop, that essentially means that the fluid is flowing too fast, that we're not able to reabsorb sodium fast enough. And so because of that, we need to slow down blood flow. So if you get increased pressure, again, that's going to speed up fluid flow through here. And as that flow rate increases, we can't reabsorb sodium. And so these macula densa cells then detect that, hey, we haven't reabsorbed enough sodium because the concentration of sodium in this fluid on the ascending side is too high. And so what they will do then is to release a vasoconstrictor chemical. So that vasoconstrictor chemical is then going to move through these mesanglial cells into this underlying smooth muscle, and then that's going to constrict the afferent arteriole to slow down blood flow as well. So both of these two are going to slow down blood flow, or are going to reduce pressure in the glomerulus to decrease glomerular filtration rate. So that's their job. The extrinsic controls, so the sympathetic nervous system, remember that the sympathetic nervous system is fight or flight. And so one of the things that we do whenever we initiate our fight or flight response, and the other thing about that is we initiate that response with the onset of exercise as well. So when you take off down the road, that is going to stimulate the sympathetic nervous system. And so the sympathetic nervous system, as part of its response to the onset of exercise, is going to cause systemic, so throughout the body, vasoconstriction, closing down blood vessels throughout the body. And so what that does is it directs blood flow away from the capillaries in the small intestines and away from capillaries in the kidneys and the liver. So remember I said at rest that the kidneys get about 25% of cardiac output. That's going to drop substantially during exercise. Basically, during exercise, we can filter the blood later. We can digest later in the case of the blood flow to the small intestines, because right now we need to direct blood flow to the skin surface to get rid of heat, and we need to direct blood flow to the working muscles so that we can supply oxygen to allow those muscles to generate ATP. So the sympathetic nervous system then is going to cause vasoconstriction here to decrease blood flow into the glomerulus so that we essentially try to hang on to as much fluid as we can. So the sympathetic nervous system is going to decrease the glomerular filtration rate to try to hang on to fluid. The other mechanism that we have, so remember these little granular cells here. So these granular cells have in them, trying to think of the vesicles. They have little vesicles that have in them renin. And so renin then is going to be released by them into the blood, which then interacts with a plasma protein that's created by the liver, which is called angiotensin 1. And so renin stimulates the conversion of angiotensin to angiotensin 1, which is then converted by another enzyme to angiotensin 2. Why all that matters is that angiotensin 2 stimulates the adrenal cortex, so your adrenal glands, to secrete aldosterone. And what aldosterone does is it helps the kidneys to reabsorb sodium. And so when the kidneys reabsorb sodium, they pull water with it. So by doing that, that's going to help us hang on to fluid volume. And then the other thing that angiotensin does, or specifically angiotensin 2, is that it's going to cause the posterior pituitary gland, which is in your brain, to release antidiuretic hormone. And what antidiuretic hormone is going to do is cause increased water absorption in the kidneys, so in those distal tubules specifically of the kidneys. So the renin angiotensin aldosterone mechanism, what it does is in response to low pressure, so decreased filtration rate, it is going to cause us to hang on to fluid. So it causes the secretion of aldosterone, which causes us to hang on to sodium or reabsorb sodium in the distal tubules, causes us to reabsorb, not aldosterone doesn't. What I want you to know about aldosterone causes the reabsorption of sodium. And then in addition to that, angiotensin causes the release of antidiuretic hormone, which then causes the reabsorption of water. So aldosterone and aldosterone and antidiuretic hormone, there's a lot of A's here. Aldosterone and antidiuretic hormone work together, with aldosterone reabsorbing sodium and it pulls water with it. Antidiuretic hormone just directly causes the reabsorption of water in those distal tubules and also in the collecting ducts. All right, so as mentioned earlier, we have to reabsorb most of that filtrate. So filtrate actually happens primarily in the proximal convoluted tubule, and then you get some additional reabsorption on both the descending, ascending loops, and then also in the distal convoluted tubule. But one of the things I want you to know is that the vast majority of reabsorption occurs in the proximal convoluted tubule. So there's two ways for things that are being reabsorbed to go. Sorry about that. Somebody came and rang the doorbell and the dog lost his mind. All right, so in terms of reabsorption, so I was talking a second ago about most of it happens in the proximal convoluted tubules. So two different ways that the reabsorption can occur. So first is transcellular, and so what that means is whatever the solute or solvent, but primarily the solute, so things like sodium, is moving through the cell. So when reabsorption is transcellular, something's moving directly across the cell. So it's moving from the apical surface through the cell, through the basement or basal surface, and then into the extracellular fluid, and then into the blood vessel eventually. So that's transcellular. So something moves from that tubule across one of the cells lining the tubule, and then into the underlying extracellular fluid. And then paracellular is when something moves between the cells. And so you can see that here, the paracellular route. So for the most part, so you got tight junctions here. So for the most part in these tubules, you have tight junctions between these cells, which would then mean that we're not going to allow things to pass between the cells. But in parts of the proximal convoluted tubule, those tight junctions are fairly weak or non-existent. And so in that proximal convoluted tubule then, we allow the passage of some water, but also calcium, magnesium, potassium, and sodium to pass via this paracellular route. In the case of sodium, so the movement of sodium is transcellular, meaning that we absorb sodium on the apical surface of these cells, and then we move it directly through the cell and then pump it out the bottom. So it moves across the cells, and then the absorption of sodium is also active, meaning that it requires energy. Because what happens is that on the basolateral surface, so down here, we're actually going to use ATP to kick sodium out. So we want to maintain a low sodium concentration inside of the cells so that sodium wants to move from the filtrate into this tubule cell. So sodium wants to move here because there's a low concentration inside of the cell. So we've created a concentration gradient by actively pumping it out the basolateral surface. And so by doing that, again, so sodium wants to move in. One of the things that we're going to do, as you'll see here in a second, is we're going to use that concentration gradient that we've created to help us pull other things inside of the cell. So things like glucose can move with sodium so that we can reabsorb them. So creating that gradient for sodium helps us do other work. So that's going to be an important function of that. There we go. Oh. So yeah, so there's that graphic. So that's us pumping sodium out, and then us bringing things like glucose, individual amino acids, and then some ions and vitamins with us. They're going to be co-transported with our sodium here through this little tubule cell. All right, so how do we reabsorb other stuff? Well, I kind of just mentioned it. So a lot of the other things that we do, the other things that we absorb through secondary active transport, which again is another throwback to chapter 3. So the primary active transport was us actually pumping the sodium out of that tubular cell. And again, by doing that, we create a gradient where there's a low concentration of sodium inside of the cell, but a relatively high concentration of sodium in the lumen of that tubule. And so then that means that sodium wants to move inside of the tubule cells. And so when it does, we let it take things with it, like glucose, like I said, amino acids, and then some of the ions. And then as sodium moves from the lumen inside of, as sodium moves from the lumen inside of the tubule cells, that's going to also pull water with it. And so that helps in the reabsorption of water. In terms of transport maximum, because there are these proteins on the apical surface of these tubular cells that actually have to allow or facilitate the movement of things like glucose from the lumen inside of the cell, there's only so many spaces, or there's only so much that we can transport inside of these cells in a certain period of time. So it's kind of like one of the other textbooks I've used the analogy of like an escalator. You can only transport a certain amount of people in a certain amount of time on an escalator. There's only so many steps available, right? And so the same concept here. There's only so many transporters available. So if we have something like lots of glucose, because normally glucose is something that we absorb, but if you have somebody who, and you usually see this in diabetes, but they have really, really high blood sugar, they're going to have lots of sugar out here in the lumen of the tubule, and so we want to reabsorb it, but we only have so many transporters, and so eventually we can't reabsorb all of it, and so the rest of it gets lost as urine. And so somebody who has uncontrolled diabetes and really high blood sugar, they're going to have extra high glucose in their urine because basically they couldn't reabsorb all of it. There was so much in that filtrate that it saturated the transporters, and so we couldn't, or they couldn't reabsorb all of it, which is what leads to then that high glucose concentration in the urine. All right. So in terms of reabsorption of the tubules, so here's your little capsule over here. This is our cartoon of our proximal convoluted tubule. Here is the loop. And then here is the ascending part of the loop, and then the distal convoluted tubule over here. So as mentioned a second ago, so the proximal convoluted tubule is the most active reabsorption site. So about 65% of the reabsorption, which you can see here, that occurs is going to happen. So almost 2/3 of it happens in that proximal convoluted tubule. We reabsorb water. And again, part of it's because of those, the junctions aren't quite as tight between those cells. And so it allows that paracellular movement. So water and calcium, et cetera, to actually move between the cells. So one of the other things here, and another throwback to chapter 3, is the concept of an aquaporin. And so what an aquaporin is a channel in the cell's membrane that allows the free passage of water. And so what we're going to get, in addition to that paracellular movement in the proximal convoluted tubule because of the lack of those tight junctions, the cells in the proximal convoluted tubule and then also on the descending loop have aquaporins in them, and so that is going to allow for the free movement of water. And so we're going to reabsorb water from the filtrate. So that one of the things that was kind of confusing to me looking at these graphics at first. So anything in the tube is going to be excreted as urine. So in terms of orienting yourself to this. So when water moves out of the tube, that means that we're reabsorbing it. So same thing here. So when water's moving out, we're reabsorbing it. So in the proximal tubule and in the descending loop, we've got aquaporins, and so we're going to reabsorb most of our water through there. And so what ends up happening, I won't get into those last couple slides. We're actually going to end a little bit before those. But one of the things that's going to happen here is we're going to reabsorb water, and so the filtrate gets increasingly concentrated here. So as the water leaves, what's left, a lot of our ions and our glucose, is going to get increasingly concentrated as we go through here. And then we're going to reabsorb a lot of those things up here in the distal tubule. And so one of the things that you can see we do in the distal tubule is a sort of fine-tuning of that filtrate. So by the time we get to the distal tubule, we have reabsorbed the vast majority of what we're going to reabsorb. So we've reabsorbed everything but about 10% of the sodium and about 25% of the water. So viewed another way, we've reabsorbed about 90% of the sodium by the time we get over here, and we've reabsorbed about 75% of the water by the time we get over there. So in terms of fine-tuning, we do that through hormones. So I mentioned antidiuretic hormone a minute ago, and so antidiuretic hormone, what it does, so normally on this side, so on the ascending limb and then the distal convoluted tubule, normally there's not aquaporins over there. But one of the things that antidiuretic hormone can do is to cause the movement of aquaporins into the membranes of those cells. So if I can back up a couple slides, this will work. So those cells, those tubule cells, are going to have little vesicles in them that'll have aquaporins. And so in response to a hormone like antidiuretic hormone, it's going to start this signaling cascade in the cell that'll cause the movement of those aquaporins to the apical surface of those cells so that they can reabsorb the water. So in response to antidiuretic hormone, then we're going to reabsorb water here in that distal tubule, but also in the collecting duct. So again, we release antidiuretic hormone. That's something that gets released during exercise, so like during endurance exercise, because if you're going to be exercising, you want to hang on to your plasma volume so that you can give it up as sweat, right? You don't want to be giving it up as urine, but instead, you'd rather give it up as sweat so that we can help be productive, we can cool the body down. So antidiuretic hormone is going to be secreted during exercise, as is aldosterone. And so aldosterone has a similar function here. So particularly in the collecting ducts, it is going to cause the movement of sodium and potassium channels to move from the inside surface of those cells to the apical surface so that they can reabsorb sodium and potassium. And so you can see that here. So regulated reabsorption of water and sodium, water by antidiuretic hormone, sodium by aldosterone. The other one on here, ANP. So ANP is secreted by the atrium of the heart, specifically of the right atrium, in response to high blood pressure. And so if blood pressure is too high, one of the ways that we can bring it down is by excreting extra fluid. And so what ANP does then is inhibits the reabsorption of sodium. And so by reducing the reabsorption of sodium, then we can, it's going to pull water with it. So what that means, we're going to leave more sodium here in the filtrate, water stays with it, so then we excrete more fluid. So that's what ANP does. So ANP helps us to, or viewed another way, ANP keeps us from reabsorbing sodium, which then causes us to excrete more water, which then reduces blood volume, which then reduces blood pressure. So that's what ANP does. Parathyroid hormone has a similar effect to aldosterone, except that parathyroid hormone plays a role in the reabsorption of calcium. So remember parathyroid hormone, we talked about it actually in 201 with the bones chapter. So parathyroid hormone, what it does, plays a really important role in maintenance of blood calcium. So blood calcium can't get too high, can't get too low, because calcium plays a number of important functions, from regulating heart rhythm to allowing for the release of neurotransmitters. So we have to have a certain amount of calcium in the blood all the time to allow for that type of homeostasis. And so if blood calcium gets too low, the primary function of parathyroid hormone is to stimulate the osteoclasts in bones to break them down and release that freed up calcium from the bones. But in addition to that, parathyroid hormone affects the kidneys to try to hang on to any of that calcium that we might have otherwise excreted. So it basically tries to free up more calcium from the bones, and it tries to conserve any calcium that we would have otherwise gotten rid of. So I left this one on here as just a graphic for you to check out in case you don't have the textbook. It's a pretty useful graphic for how we maintain blood pressure. All right, and then secretion. So secretion helps us get rid of things that we don't want. So you can see there happens largely just like absorption does. Secretion happens largely in the proximal convoluted tubule, that's the PCTs. And so that is, again, the intentional movement of certain things into the filtrate. So helps us get rid of drugs. So for example, we get rid of non-steroidal anti-inflammatories, so things like ibuprofen are cleared by the kidneys, and so we clear them by secretion into the filtrate. But in addition to that, we get rid of hydrogen ions, so that's how we're going to maintain our blood pH. We get rid of potassium, and then also ammonium and creatinine this way. So on an earlier slide, I said I had my list. That was my list. So those are the things that we primarily secrete through, or we primarily get rid of through secretion. Also plays a role in getting rid of urea. About half of the urea that we eliminate, we actually do that through secretion. And then, as mentioned, get rid of potassium and then get rid of hydrogen to control our blood pH. The countercurrent multiplier and exchanger I won't get into because I kind of got into the important parts a second ago, which is just that, so on the descending side, you get rid of water over here, so you end up with this really concentrated filtrate here, and then we get rid of sodium over here to balance the osmolarity back out, but I won't ask you anything about this. So don't worry about anything about the countercurrent multiplier exchanger. Don't worry about any of that stuff. So then, that brings us to the end at just over an hour. So one of the things I would tell you to make sure that you do is to look over the diagrams of the kidneys on the anatomy app. So be sure to check those out. You can, on the full body, and I should actually probably just show you. So on the full body, you can dissect a little bit, and so then you can look at the lobes, you can look at the renal pelvis, the blood vessels, all those kinds of things. Mostly I'm interested that you're able to identify like the cortex versus the medulla. And then the other thing would be that there is a microscopic anatomy of the kidney. And so make sure you check that out as well. So if we go to our primary model here, it'll allow me to, if I go to the urogenital, it'll allow me to give him some kidneys, which I will do. There's his kidneys. I'm not going to restore. And so if we zoom in on his kidneys, one of the things that you can do is to view the inside of them. So you can hide that, right? So we can hide this here. And so then you can see the cortex, the medulla, the columns. You can see the pelvis, the calyces, the major calyces, the minor calyces, etc. So just before the next test, make sure you look over this so that if there's a picture that looks like this with these guys highlighted, that you would be able to identify that those are the columns. Because there's a few questions in the test bank that look like that. The other thing that I would like you to look at is to make sure you check out the kidney lobe over here. And so then on the lobe, so obviously you've got your cortex here, your medulla here. So there's our pyramid. Sorry. So there's our pyramid there. There's our columns. Okay, and so then the other thing from there, make sure you're able to identify, so there's our little glomerulus, there's our capsule, there's our proximal convoluted tubule, there's our descending loop, ascending loop over here, distal convoluted tubule, okay? And then so just kind of run through them like that. So that if there's a picture, because there are some in the test bank, where it's just one of these things highlighted, so let's click on the, not that. There we go. So the distal convoluted tubule, so if that's highlighted on a picture, just make sure you're able to identify it. And then that's it. So then that takes us through all the material for test 2. So good luck on that, and we'll see you next week.